So, hello everyone. Thanks for joining us at this webinar tonight. My name's Ria Cohn, and I'm the Director of Conservation at the Swaner Preserve and Eco Center, and I use she, her pronouns. Our mission here at Swaner is to preserve, educate, and nurture both in our community. So tonight's talk is part of Swaner's Walks, Talks, and Workshops series, designed to help keep everyone engaged in nature and the world around us. And a couple of kind of housekeeping things while we're here tonight. I'll introduce our speaker in just a minute, but we also have Hunter on here tonight, Swaner's Director of Visitor Experience and Exhibits, and she's kind of running the back end. So if folks have any technical questions or things like that, Hunter will be helping out there. Your camera and sound are off as participants, so don't worry if you're making dinner or doing anything else. There'll be an opportunity if you want to be unmuted to ask a question later. I want to thank Summit County's RAP Tax for supporting Swaner's Walks, Talks, and Workshops series. And after this webinar, you'll receive an email from me that will include a survey, and that is super helpful for us to determine what our community wants to see. What kind of topics are you interested in, and learning more about? So we'd love if you could take the time to fill that out, and we can better serve the folks that are here tonight. It will also contain any helpful links that we talk about tonight, so don't worry about typing anything, writing anything down, or information that we discuss. So I'll send that along as well. And this webinar is also being recorded, and that recording will be sent to participants if you have to step off, or if you're watching this later and you weren't able to attend live. There will be a Q&A towards the end of this webinar. If you have a question that is related to what we're talking about right now, or a clarifying question, throw that in the chat or the Q&A. Those are both options for you to put your questions in, and myself or Dr. Osborne can answer those in real time. Otherwise, we'll have a Q&A at the end, and kind of see what questions and comments folks have. And that will be an opportunity where we can unmute you at that time if you want to ask your question yourself. But a couple of just upcoming Swaner things. It is fall, maybe winter as of this weekend, we will see. And our new in-house exhibit, Wetland Supermarket, is opening Friday, November 28th. Really excited for that. And Hunter's been working super hard on that. Park City's annual Christmas bird count will take place Sunday, December 14th. And more information will be posted on our website in just a few days on that. And then the Eco Center will also host the Wild Winter Art Market on Friday, December 19th from 5 to 8. We have an awesome lineup of artists and items like prints, ceramics, jewelry, more. You can see what artists will be there, all nature themed and support Swaner. I'll send some links to those in the follow-up email. But busy season as we shift into winter. But tonight's talk highlights drylands, which cover 40% of the Western US and are sometimes dismissed as wastelands. But these systems and landscapes are understudied and commonly misunderstood, and they support billions of people, store vast amounts of soil carbon, and are predicted to be highly sensitive to intensifying changes in climate and land use. So we're very excited tonight to have our speaker, Dr. Brooke Osborne, who is an assistant professor in the Department of Environment and Society based at USU's Moab campus. She's an expert in biogeochemistry, the science of how ecosystems work, and strives to advance our understanding of how terrestrial ecosystems respond to and shape our changing world. And she's especially fascinated by drylands. So I'll turn it over to Brooke. Thank you so much for being here tonight.
Awesome. All right. Thanks, Ria. I'm going to share my screen and see if it works as well as it did when we practiced it. All right. So I assume you can see just the slides and not the whole presenter mode, right?
Looks good.
Okay. All right. Awesome. Well, thank you everybody so much for being here today. Like Ria said, my name is Brooke Osborne. I'm an assistant professor at Utah State. I'm based here on the beautiful Moab campus, which I highly recommend visiting if you ever have the opportunity. And I'm a biogeochemist and an ecosystem ecologist. My goal here today is really to help you walk away with a fresh perspective on drylands and how they impact our lives in the world. And so this title here, the sneaky powerhouse, the carbon cycle, right? That's kind of cute, but also a little bit vague. What I most want to share with you today is this area of research that's been keeping me up at night for a while now. In the last couple of years, I've really focused almost all of my work on this larger question of how can we better understand and more responsibly manage dryland soil carbon in a time of rapid change, right? We're living through a time of rapid change where we have overlapping climate changes, land use changes, and these raise a lot of questions for the dynamics of ecosystems like drylands. And I'm in the early days of this effort, so rather than throw a whole lot of results at you, I'm going to discuss broadly why this matters and what we're doing about it in my lab right now. Also, here's our outline for the talk, but I do want to reiterate what Ria said, which is if you have questions in real time, I would love them. Please interrupt anytime. It's always odd talking into black squares, so please reach out if you have any questions or I can clarify anything as I go along. I want to start out just by introducing my field of science, which can take a little bit of explaining. And I don't know if any of you are familiar with biogeochemistry, but if you're not, I joke possibly too often that it sounds a little bit like there's three scientists stacked up in a trench coat, right? What does this mean? You're sort of smushing all these sciences together. But actually accurate analogy for biogeochemistry would be a multi-tool or a Swiss Army knife, right? It's a set of tools that we have to use in tandem if we want to pick apart the complexity of how ecosystems work. And to break that kind of complexity apart, you have to pull key tools and concepts from the broad range of fields, biology, geology, and chemistry, but also physics and many others. And so when you think about biogeochemistry, you can think of it like a multi-tool for exploring how complex ecosystems work and change. One of the key ways that we apply this tool in order to explore complicated ecosystems is by distilling them down into pools and fluxes that we are able to measure. Some of you maybe have seen a figure like this at some point in your life. Maybe it's giving you flashbacks to a textbook in grade school. Let me see if I can get this tricky pointer working. Okay, so what this is doing is illustrating the carbon cycle at the scale of an individual tree, and it's also a good visualization of how my brain works all the time. So here you can see carbon fluxes coming from the atmosphere in the form of carbon dioxide into the biomass of this tree through the process of photosynthesis. Carbon is also leaving the biomass of the tree and re-entering the atmosphere through the process of respiration. So in these examples, the pools would be the vegetation biomass in the atmosphere, and the fluxes would be these processes of photosynthesis and respiration. And so you can imagine how ultimately the amount of carbon and the form that it's stored in that exists in the atmosphere, in vegetation, in soil, in rock, right? These are the net effects of many, many biological, chemical, physical, geological processes that all work together to shape the way an ecosystem functions. So takeaway here, biogeochemistry, it's a tool. It lets us tease apart complicated ecosystems into quantifiable pools and fluxes. Another reason that I'm really obsessed with biogeochemistry is that many of the most compelling and challenging environmental issues that we face today really are questions about biogeochemistry at their very core. So climate change, for example, maybe you've heard of it, is due in large part to human alterations to the global carbon cycle. So here we've scaled way up from that individual tree, and we're now looking at the carbon pools and fluxes worldwide. And I don't expect you to read these as tiny. They're probably also very outdated because this is now from IPCC in 2013. But the point here really is this red text and these red arrows. What those are representing in this diagram is how humans have modified the global carbon cycle and contributed to climate change. So in addition to thinking about how that pristine tree and a meadow functions, right, we can use biogeochemistry to better understand how ecosystems interact with humans, and not only how we're contributing to problems, but also to identify potential avenues for solutions and even management strategies to address those issues. We use this approach across all different spatial scales and many different ecosystem types. Here, I'm going to be focusing specifically on the terrestrial biome. So imagine with me, if you will, this little cartoon cloud represents all of the carbon dioxide that's present in Earth's atmosphere. This arrow that I'm showing you coming in from the right is representing all the carbon dioxide entering the atmosphere through the combustion of fossil fuels, which, as we hear in the media and read and understand, is a major driver of climate change, right? And that is absolutely true. But what you may not think about as much is this arrow that I've just added to the left, which, by the way, is drawn to scale. And this represents carbon dioxide entering the atmosphere from the terrestrial biosphere. So this is the respiration of plants and microorganisms. And at first it's like, yikes, right? Like that seems scary. Until we remember that this is a natural flux and therefore it has a counterflux, which is photosynthesis. So in a perfect world, these fluxes remain balanced in moving carbon through pools, and the effect of the terrestrial biosphere on atmospheric CO2 in this particular scenario would be neutral. But what happens if the size of one of these arrows change, just gets shaved down a little bit or increased a little bit? Maybe it's a shift in the frequency and severity of wildfires. Maybe it's the type of vegetation cover that there is, right? There could be enormous consequences to even small changes in these massive global scale fluxes. So we can use biogeochemistry to study how global change, things like wildfires, plant cover type shifts, climate change, can influence the sizes of these arrows. All right, so we've talked a little bit about what biogeochemistry is and how we use it. Now I want to talk about why dryland biogeochemistry matters so much, and then specifically why I'm obsessed with soil carbon in dryland systems. So why do we care about dryland biogeochemistry? I was going to ask you guys that, and we can chat about it. I know it's hard in this format, so maybe you can think about it to yourself a little bit. Why would it be that we care about how dryland ecosystems function? And, you know, it's perfectly reasonable to say that we care because they're beautiful or because they hold profound cultural significance, or we just want to know, right? Basic science is a very non-trivial motivator to care about how a system works. But what I'm going to really highlight right now are some of the specific reasons we're motivated to study dryland biogeochemistry in the context of global change. So not just how they work, but how they're changing around us and what that means for the future. One reason we care, right, is drylands are absolutely gigantic. So this map of the world is showing in these different colors where different types of drylands are located around the world. And like Ria said in the intro, they cover something like 43% of land area on Earth right now. That number is predicted to be more than 50% by the end of the century because of climate change. And so intuitively, it's not hard to imagine, just looking at the scale of these places, why dryland ecosystem functions matter, right? So they're huge. They also are very important. They're home to more than 2.5 billion people. Those people's livelihoods rely often on provisioning ecosystem services like the production of fiber or food. In fact, about 60% of all food production on Earth comes out of drylands. And they also play very important roles in global biogeochemical cycles, and in particular, the carbon cycle, which we're going to talk more about soon. But in a nutshell, they are dominant drivers of net primary productivity, which is essentially the rate at which energy is stored in the biomass of plants. They are dominant drivers of short and long-term trends in the quantity of carbon in Earth versus in the atmosphere, and they store about 40% of all soil carbon on Earth. So we care about biogeochemistry because they are huge, they are globally important for many reasons, and they differ in fundamental ways from mesic systems, meaning it's very tough to translate our relatively deep knowledge about wetter places, like this meadow on the left, to our understanding and management of drylands, like over here in Moab, where my dog's on a hike. And there are a lot of things that make drylands different. We could talk about it all day, and if anybody has questions, it sounds great to me. But one key and sort of intuitive example is that they're patchy. So what you're looking at here is a really common scene down here in Southeast Utah, right? You see this sort of bare soil that we all know that there's biological soil crust there, but relatively bare soil that's dotted with vegetation like these shrubs that I've circled. And you can very quickly see, right, that even if there is some kind of light or early crust development here on the soil, there's not much happening. And under here, boom, right, you've got these thick moss mats, you've got different plants growing. So just within inches of each other, the biogeochemical cycling in this system is very, very different. And that's because drylands have pretty low resource availability. Yes, they have low water, of course, but they also have low concentrations of carbon and nutrients. And that means that even the small differences afforded by the presence of this shrub in terms of protection from ultraviolet rays, a decrease in temperature, a higher propensity to hang on to soil moisture beneath that plant, support the presence or absence of vegetation and drive majorly big differences in biogeochemistry. So this patchiness is very, very interesting to folks like me, and hopefully to you as well. But it's also a reason biogeochemistry in drylands is really tough to generalize and model. So what do we say so far? They're huge, they're important, they're different than the systems we understand well, and they're also predicted to be particularly vulnerable to climate and land use change. And this is due in part to that low resource availability I was just talking about, and has a similar dynamic to that really localized scale we were just discussing, where even small changes in mean annual precipitation or temperature or land use practices, like the intensity at which we graze drylands, can have really outsized consequences. And there's no question that drylands are experiencing their fair share of changes. Climate projections predict that drylands are going to get warmer and drier faster than other biomes. And we're already seeing strong evidence of these changes in the Western US and all around the world, including this lovely mega drought that we're currently living through. So I'm going to use Southeast Utah and this Four Corners region as an example. This map comes from NASA a couple of years ago. It's showing the intensity of the mega drought increasing to this ominous sort of purple color. And you can see, right, that our region is just getting hit especially hard. And it's true. We're getting warmer and drier, and it's troubling for a lot of reasons, not least of which is that we really play a critical role as a water source for the entire southwestern US. But yes, Southeast Utah is really a bullseye for increasing temperature and drought across the country. And on top of these rapidly changing climate conditions, this region and drylands around the world have this layered challenge of land use changes with lots of different priorities at play and practices. So just to stick with our Colorado Plateau example, oil and gas extraction, there are more than 90,000 extraction sites currently on the Colorado Plateau. It's a lot, 90,000. You can see it while you're flying over, you can see it from space. Here we have grazing, so about 90% of the Colorado Plateau is open to livestock grazing, right? 90%. And that list goes on, and they don't include in an isolated way. They all interact with each other and changes in climate. And we're seeing those consequences. Things aren't great. So sticking with the American Southwest, these are maybe some images or stories that you've heard about in recent years. And so drylands are expected to be sensitive and vulnerable, and we're seeing that play out. And yet, we know a lot less about the consequences of global change in drylands than we do in wetter ecosystems. Just as one example of that, there's this repository of data, soil respiration data. So that means the carbon that is leaving the soil and entering the atmosphere through the flux of respiration of microorganisms. So basically bacteria breath right in the soil. And of all those data points from all around the world, only about 5 to 10% of those come from drylands, even though they cover 45% of Earth's land surface. So there's lots of possible reasons for this. It could stem from a really common misconception that drylands are these unproductive wastelands, right? But whatever the reason is, there is a fair amount of catching up to be done. All right, so we've been talking about why dryland biogeochemistry matters. Drylands are gigantic. They cover a massive portion of our planet. They are extremely important for humans and for natural biogeochemical cycles at local and global scales. They are very different from the ecosystems that we understand well. We can't just use that information that we have. They tend to be vulnerable to things like climate and land use change, and they're also understudied. So Gidvu, right? It's a horrific mnemonic device, but there you have it. It's the best I could do. All right, so why then is dryland soil carbon cycling particularly important? So now let's take a narrower look into that. First up, soils are a hugely important component of the global carbon cycle. Here I've just separated out the totals of pools in the atmosphere in vegetation and soils in petagrams of carbon. Soils contain more carbon than plants in the atmosphere put together, right? So they are a massive player in the global carbon cycle. And if you take that huge piece of the pie and look just at drylands, drylands contain a massive share of that, about 30% of total soil organic carbon and 80% of total soil inorganic carbon for about 40% of all the soil carbon on Earth, we think. And that can feel a little bit unintuitive when you think about a dryland compared to another ecosystem, right? So what about a tropical forest, right? If I had in front of you magically a square meter of a dense, lowland tropical forest in Costa Rica, and right next to that was a square meter of sand flats in Moab, Utah, right? And you were asked which one to study about carbon cycling, you're going to look at the tropical forest, right? There's more carbon, it's cycling faster. That's absolutely true. And also one reason tropical forests are so glamorous, right? They received a lot more research attention regarding carbon cycling. Drylands are very much the Jan of this particular biome. But what drylands lack in resources and productivity, they make up for in size, right? Remember Gidvu? I don't know how you could forget. It's so memorable. But drylands are huge. So even though carbon density is really low, the area is massive. And that is how we come up with this 40% of soil carbon. Because of these massive stocks of carbon, dryland soil carbon cycling is really important for the global carbon cycle and climate change. We get it. But I also want you to remember that soil carbon serves a lot of other critical functions than just influencing climate change. And some of those you can very easily see with your eyes. So this soil that's on your left has more organic matter in it than the soil on the right. And if I am growing plants, right, I want to grow it in the soil that's on the left. There's higher levels of fertility, a greater ability to hang on to water. And variables like that obviously directly impact our ability to grow crops, to graze cattle, to have clean air and water, things that we really like. So I just want to recognize carbon as a valuable resource beyond its role in climate change as well, but something that benefits folks locally. And yet we lack even really basic information about these major stores of carbon, like exactly how much is there, right? 40% on Earth. Okay. Where is it? Is it in the surface? Is it down deep? What forms is it in? How quickly is it turning over? What does it look like across different landscapes? And if you ask a biogeochemist that, like, we really don't know. We're still at square one compared to these Marsha tropical forests, right, who have received all this funding and attention and time. The tides are shifting. There's a lot of great momentum in asking these questions, but even those really foundational ones, we don't have good answers to. And that's why I pace around and I worry about how we can improve our understanding and our ability to manage soil carbon in drylands under so much growing pressure from global change. So what are we doing about it, right? What am I going to do? Well, I just want to wrap up by highlighting a couple of my lab's ongoing projects. Like I mentioned, these are still pretty early days, but I'm really excited to share our ideas and momentum and to hopefully talk about them with you. This first project, it's a partnership with the Utah State Office of the Bureau of Land Management, who actually approached us to help lead a program exploring soil carbon distributions across Utah's drylands and the impacts of grazing in particular on that carbon. That is, wow, that's really exciting. And the reason that's so exciting, even though there's been a lot of attention paid broadly to soil carbon recently, is that data to support carbon management and policy on public lands are very rare. And so it's cool that the BLM is thinking in this direction. One reason those data are rare is that it's really challenging work, right? And it requires an incredible team of really complementary skills. And we've been able to round one of those up. I just wanted to share our full lineup here. so you can see their faces. But just for the sake of time, I'm going to focus on work that's being led at USU, specifically by my former PhD student, Savannah Adkins, now Dr. Adkins, in close partnership with Dr. Sasha Reed from the US Geological Survey, Jason Burgess-Conforti at the Bureau of Land Management, and Dr. Rory O'Connor at the US Department of Agriculture. And we came in motivated by this piece of the larger question. So how does soil carbon vary with grazing intensity in drylands of southeast Utah? And Savannah really started by developing a series of hypotheses, which are best summarized and maybe simplified in this particular figure that she's made. So up here, she has this arrow of increasing grazing intensity, low, medium, and high, in case you can't read it. And her general prediction was that there'd be a sweet spot, that moderate grazing would support the largest pools of soil organic matter, shown here in this little blue square, because a lot of these systems that we're talking about evolved alongside natural grazers. And plant productivity may therefore be optimized with some grading because they've, again, evolved in tandem. And that increase in biomass also increases carbon inputs to the soil through processes like decomposition of dead biomass. The idea was that too little grazing, you wouldn't have that extra stimulation of plant growth, and too much grazing, you just can't recover and soil carbon drops. Essentially, she proposed a Goldilocks situation. And so we designed a field sampling campaign to test her hypotheses. This particular effort was focused on Grand County in Utah. I outlined down here in red. And those background orange and yellow and red colors are showing the different groups of drylands, which make up a tremendous amount of our state. And these little blue polygons layered on top represent grazing allotments for the Bureau of Land Management, which covers about 42% of our state. So this is not a trivial question when we think about the well-being of the ecosystems that we live in and use and enjoy. And to identify our particular sites, we combine multiple data sets. And I'm going to quickly step through this. If you have questions, I'd be really happy to come back to it. But here on the left, I'm showing Bureau of Land Management grazing permits for Grand County in animal unit months, AUMs. And grazing intensity increases from blue to red within individual allotments. And this is not actual grazing. Unfortunately, cows don't go out and evenly graze everything in an allotment, which really causes a lot of people, for folks who have questions about grazing and biogeochemistry. So you can think about this more like the potential grazing, maximum grazing that could possibly legally take place on an allotment is shown here. Then we compared that to, or essentially took a ratio of that with above ground biomass. And so this map comes from RAPS, so Rangeland Analysis Platform, which combines satellite imagery with on-the-ground measurements collected by the Bureau of Land Management, where those field data are used to train RAP to convert net primary productivity into above ground biomass. So this is pounds of biomass per acre. And so, like I said, we basically took a ratio of these two things to create a map of what we call usage classes, labeled here as low, intermediate, and high. And so if that ratio was between 0 to 0.5, that ranges everywhere from no grazing to a take half, leave half type scenario. So that's low. Intermediate, 0.5 to 1, you go from your take half, leave half to take all, leave none, consuming all net forage growth for that year. In a high scenario above 1, that means that more above ground biomass is consumed than replenished each year. So there's a decrease over time in the amount of biomass in that site. And we identified about 20 sites in each of these different classes, and we selected those to cover a number of other key environmental variables we wanted to consider in terms of soil carbon. For example, we spread them as evenly as we could across vegetation classes because drylands are super diverse and often include everything from grasslands to shrublands to wetlands, woodlands, not wetlands. And we know that carbon cycles differently within each of these system types. So we had high, medium, and low grazing intensity across each of these vegetation classes. We also know that vegetation in drylands is really patchy, and so we wanted to capture that heterogeneity as well. And we sampled beneath dominant shrub and perennial grass species, as well as interspaces. And a lot of these sites we also collected from really mature patches of biological soil crust or even under trees if they were present to try and get a clearer and more complete snapshot of those sites. And then at each of these locations, we went out with our handy gas-powered soil core and collected soil cores of up to a meter deep, or until we just couldn't get any deeper. And in this way, we're able to learn about soil carbon content across a wide range of vegetation categories, grazing intensities, and microhabitats. This is what one of these cores look like when you pull it out of the ground. You can see there's a lot of just visually differences in the soil as you get deeper. And we cut each of these a number of times into set depth increments so that we could look with relatively high resolution at how things changed with depth. So not just surface soils, but also subsurface soils. And I gotta tell you, we recently wrapped up the processing and analysis of all this, and we're wading through the data right now, and it's a tremendous amount of data. Really impressive data set that Savannah was able to organize and pull together. And I'm not going to go down that rabbit hole, though I'll answer any questions you guys have, but just wanted to share one sort of interesting snapshot of some preliminary findings. So here we have three figures. We have TOC, total organic carbon, POMC, particulate organic matter carbon, and MAOMC, mineral associated organic matter carbon. So all you need to know about this right now is this is all the organic carbon, and these are two different fractions that we often break soil carbon out into to tell us more about how rapidly it might turn over versus be stabilized in the soil. And the key difference that she has found so far is that in these grazing intensities, low, medium, high, where low is red, medium is green, high is blue, there is lower concentrations of soil carbon in the low grazing intensity, and soil carbon actually increases in the medium and high grazing intensity, which is very interesting. So it aligns, a bit with her sort of Goldilocks theory, right? And I should say this is just surface carbon too. This doesn't include the deeper horizons, but this is across all those different vegetation classes, all these different microhabitats. And it's not a super strong signal, but it's there. It does seem like there's a sweet spot, but that high grazing intensity also includes more carbon than low. So this raises questions for us that we're excited to explore in our data set more deeply and future studies about what are those mechanisms driving that? Does that signal hold true as you go deeper? Is this driven primarily by shrublands, right, versus grasslands? So all these wonderful questions we can follow up based on some of these early findings. Okay, so that was one of our projects focused on grazing in public drylands of Utah. The second project I'm just going to touch on is focused on how climate and nutrient availability impact soil carbon worldwide. So first, just to pause here, why are we thinking about nutrients? We've talked about why climate really matters for soil carbon, but why nutrient availability? If you think we've screwed up the carbon cycle, you should take a look at the nitrogen cycle and also phosphorus. So we've more than doubled the amount of reactive nitrogen inputs to terrestrial ecosystems all around the world. It's not quite as dramatic with phosphorus, though we have still certainly altered that system as well. And so human activities are responsible for emissions into the atmosphere of nitrogen and phosphorus, which then enters at different rates back into ecosystems where it normally would not be. You're essentially just dumping fertilizer out into those places. And so what happens in terms of the biogeochemistry there, including soil carbon cycling. So nutrient deposition is another form of global change. And we wanted to know how climate and changes in nutrient availability mediate dryland soil carbon. And this is a big question. And we wanted to get at it in a meaningful way. And so we partnered up with a group called the Nutrient Network. So the Nutrient Network is a long-running now cooperative of nutrient addition experiments located all over the world. So these kind of coordinated networks mean we come up with a protocol, we all distribute it, and where you live or work, you set up and maintain your own site. You apply the treatments, You collect data in identical ways, and it produces data sets that one of us could never produce on our own, right? And so it's comparable data points that you can query for all kinds of information. And each one of the pins on this map represents a nutrient network site in a dryland that has existed at one time or another. You can see they span 6 continents. They cover a really wide range of environmental and climatic gradients. So to evaluate climate effects on soil carbon, we're comparing sites along these natural climatic gradients to space for time comparison. To evaluate nutrient effects on soil carbon, we're comparing samples from the long-term nutrient addition plots at each of these sites. Oh, there's our site and Arches. Probably the best one, is biased. But so each one of these sites has identical nutrient treatments applied every year. And for some of these sites, it's been 10 or 15 years at this point. So this is a long-running project. And the protocol that we use is to apply nitrogen, phosphorus, and potassium in a full factorial design. So every possible combination of those, along with a control which gets no nutrient treatments. And our work is focused specifically on nitrogen and phosphorus, which I mentioned earlier are impacted by atmospheric deposition. So that's why we're focusing on control nitrogen, phosphorus, and nitrogen with phosphorus. And so what we did was we reached out to the coordinators of all these different nutrient network sites in Drylands, and we asked them if they wanted to participate. 18 of them did. They're from South Africa, Australia, Mongolia, Portugal, all across the Western US. And then we constructed and shipped out these soil sampling kits that included PVC cores that we cut to the depth of sample we wanted and beveled, and basically it's not very high tech. You go out there with a mallet, hammer it into the ground, pull the core back out, cap both sides so you have an intact cores, and ship them back to us here in Moab. We've recently received most of these back. I think we have 16 of the 18 kits that we're expecting. And what we've done most recently is we cut each of those tubes again to have the surface and subsurface data, and are now processing, and we'll analyze those for soil carbon content. So these are just some fun recent pictures. I like this one to give you a sense of how different these soils are. This red, red, red sandy soil from Australia in comparison to this more organic matter rich, darker brown soil, and this really dried out soil next to it. So it's a great diversity of samples that we've been able to collect. Of course, shipped out many, many boxes, made really good friends at FedEx, and here we are cutting up these tubes just very recently. So by measuring soil carbon at a range of depths across diverse sites, we can really improve our understanding that those baseline questions that we talked about: where and how are dryland soils storing carbon, and then what happens under different climatic conditions and with nutrient additions. And very excitingly, we have a new lab member. This is Elizabeth Currit. She's a PhD student in my lab starting this semester, who's really taking off and running with this project. And this work is also in close collaboration with Dr. Kelsey Jensen at the Soil Inventory Project, as well as Sasha Reed at the U.S. Geological Survey. A next step for this, once we have our data from the field, those empirical data, is to help improve model representation of dryland soil carbon cycling. So, we rely on models to make predictions and decisions about the future, and without much data from drylands, or without data from drylands informing the creation of models, right, we're not doing a very good job of making those predictions in the first place. And so we're working with collaborators at Colorado State University, led by Francesca Cotrufo, and the modeling team behind a model called MEMS 2.0, a powerful new mechanistic model. And we'll use our data from this project to help validate and calibrate their model so that it can more accurately represent and predict dryland soil organic carbon cycling, which, as I mentioned, is something that models have done pretty poorly in the past. This particular model is poised to replace Sentry, which is a model that was developed in the mid-90s and is awesome and very commonly used to simulate plant soil carbon and nitrogen cycling dynamics in Earth system models, including those that groups like the IPCC would work to forecast future climate change. So this work has the potential to help create a much stronger predictive framework to forecast how drylands respond to and feedback on global change. So that's all I've got for right now. I do want to just say I've got a new student coming in, Dav Sanarud, who's already working on his project, which relates biological soil crust to soil carbon cycling and sagebrush systems. And Dr. Johnny Arteaga is a part of many of these projects as well. He's a postdoctoral fellow in our group. And he is doing some incredible work to link existing vast dryland soil carbon data from the field with remotely sensed products and get a sense of what those patterns both above and below ground look like. And so that's it. I'm happy to answer any questions you all might have.
You can hear the applause. Thank you so much,
Yeah,
Dr. Osborne. That's so fascinating. And I love the memes, of course. Folks, if you have questions, you can put them in the chat or the Q&A. But just, yeah, I think that is so fascinating. I think we hear, like you said, about wetlands and rainforests as these carbon sinks, and at Swaner, which is about 850 acres of wetlands. That's the Jan of our ecosystem, but those drylands are so important. And I think one of the coolest parts about living in the West and learning in the West. And yeah, so if folks have questions, you can throw them in the chat. I also really loved kind of the tools that you're using, and I loved that picture of all the different soil types as well. That was really cool. But I'm wondering if you talked a little bit about, you have a lot of different partners, partners that you work with globally for, especially your nutrient related project. Do you have partners that you work with globally for like dryland carbon in particular that might be interesting to hear about?
Yeah, I would say, as I mentioned, it's like definitely the Jans. Like we kind of get the shaft with dryland science a little bit. But that is changing in real time. I'd say over the last 3 to 5 years, there's been a massive increase in attention paid to these ecosystems, and it's going to be really exciting to see what kind of findings come out over the next, 3 to 5 years. A lot of folks have projects like ours that launched a couple years ago, and we're still waiting to learn what they have to say because these field-based studies can take so long. There are a lot of folks in particular based in Australia, as you might guess, also quite a few drylands there who we really enjoy working with. And then, there's groups that are more focused on grazing and rangelands within drylands. There's a tremendous amount of overlap between those two groups. And so, folks working in the federal government have a large investment in understanding that and make excellent collaborators for those of us at institutions like Utah State. And so it is a small but growing community and a very sort of vocal and enthusiastic one that I think is finally achieving our goals of making everybody pay attention to drylands. And so, yeah, things are turning around, I think, and certainly lucky to have a lot of collaborators across a lot of different spaces.
Awesome. Yeah, we'll have to get down to the new Moab campus. Sounds incredible that USU has down there, and just really interesting work coming out of there. Tom has an awesome question. That one point early on, you mentioned that grazers evolved with our drylands, but not cattle that we know today, but bison. So thinking big, what would be the benefit, if any, of if our cattle cohort were replaced with bisons? Maybe like can you describe some of those differences and maybe what we might expect to see?
That's an awesome question, Tom. And I am a soils and plant gal, and so I hesitate to say anything too definitive about what those sort of feedbacks might be between different ungulates and the plants. I imagine there are differences in what the preferences for these different organisms would be, what they would eat first. Patterns in grazing really matter. If the grazers are all concentrated together in one place versus roaming more freely. And so I really think the elements that would come into play there are not just who's grazing, is it bison or cattle, but also how free are they to roam and follow those sort of natural patterns that they might typically throughout the year in concert with vegetation. And so I think certainly there could be some key differences there. And it would be very interesting to find out. Are any such experiments underway? Tom asked if anything like that is being done right now. Um, hmm. I'm not, I'm certain that there are studies happening relating bison as well as cattle to soil carbon storage and sequestration. So when I went to biogeochemistry, I was one of these tropical scientists, right? I still am. I still do tropical stuff, but I'm very dryland biased now. And, you know, I just say that there's more interest in this stuff than I ever thought there would be. I thought it was like me and the 30 other people doing this, but because of carbon credits, like the carbon credit market, and because of grazing and all this overlap, there's kind of a frenzy right now of what do we do? What does it mean? Can we measure this? Can we look into the future? How do we award, financial benefits to folks? And so there's a lot of people sort of coming in to fill that space and a lot of motivation. And so that's just brought a lot of energy and money to questions like this about commercially grazing cattle or bison in particular. Yeah.
Wow, yeah, fascinating. Like bison are so interesting, and I mean, just doing a quick search of some work that is maybe occurring related to carbon and bison, and yeah, really, as Tom I'm sure knows and assumes, just really different, yeah, grazing patterns and impacts
And
to the earth.
I'll say one more thing. I said I'm not a total expert in this, but I also know there's some fascinating patterns east and west of the Rocky Mountains related to grazing because natural grazing was so different in those places that there are sort of fundamental differences in patterns that we see there as well in terms of relationships between herbivores and plant growth and soil carbon.
Cool. I had a question about kind of related to grazing and the data that you're using there, low intensity, high intensity. Do we have good quality data on like the actual intensity on these grazing allotments that's
Yeah,
available?
yeah, Rhea, you hit the nail on the head. So, all this sort of frenzy I'm talking about, people trying to understand relationships between grazing in particular and soil carbon, it's extremely difficult because we live in Utah, right? You all drive by these allotments and see cattle. They're not grazing things evenly. You can say, here's my allotment. You walk out there with your soil core. Where do you sample, right? To find a representative sample for that area. And then if you're going to come back and try to quantify change in soil carbon or to prove that you have stored more carbon, how do you do that, right? Do you sample in the same exact place? And so the answer is no, there's really not a good way to do it. We're all trying different ways. Some folks locally will do like a transect and count how many cow patties there are there as some kind of a concentration of cattle, or will measure different distance from, hot spots of activity like a watering hole or a shade tree. And so there's some relationships there. But we're all just kind of taking stabs at it. And what I like about this approach, it's not perfect, but it was our way of trying, can we sort of scale this up in a way that we can use tools everybody has access to and make some kind of predictions that are clear. So like, does this tell us something? Is this a tool and a metric that we can take forward? And maybe it's not, and that's okay. Like we really are in these early stages of just trying new things and figuring out and quantifying intensity of grazing is crazy hard. Folks are also into using these virtual fence collars now on cattle. And so it's just what it sounds like. You can control where the cows go and when and keep track of where they are as well. And so, some of that technology along with remote sensing is making it a bit easier, but it's still pretty hard.
Yeah, yeah. Folks might see those. There's some virtual collars. I think the cattle McPullen barn for Park City local folks have had those virtual collars, which are really interesting and total game changers that are becoming more available and more affordable for folks managing cattle. Another question we had, which maybe can of worms as well, is what role does cryptobiotic soil play in carbon storage and release?
Ooh, I'm so glad somebody asked about BioCrust. So I love BioCrust. I think that's how you guys actually tracked me down, was I made a little video about BioCrust. That's another um, topic we're very into over here in Moab. But yes, absolutely, biological soil crust, in particular in drylands, plays a major role in soil carbon storage and cycling. So just looking at that patchiness of the landscape. In some areas, it's more significant than others, but in those bare soils, in particular, they are a dominant driver of carbon and nutrient cycling. It's something like, I keep throwing out these percentages, but like 12% of Earth's surface is biocrust or something, right? That's not anything to sneeze at either. And in these landscapes where cycling overall is relatively slow and storage is low, they really are this like consistent engine cranking. And some members of these communities that make up biological soil crust photosynthesize. They help maintain the soil in place, which prevents losses of that hard won carbon through erosion. And so, that's another overlapping variable, particularly in grazing, because you have that physical disturbance of cattle coming through, or even in these oil and gas pads we're talking about, where you have trucks running around or a pad being installed, and then you lose that stability of the soil. And that may be one of their most direct impacts there, is when they're gone, that carbon in the very surface of the soil, it takes forever to accumulate, can be gone like that. And so they absolutely are key players in carbon storage and sequestration in drylands.
Very cool. I know, I mean, folks in northern Utah, Park City, huge visitors to southern Utah's deserts. And so we'll share that video in our follow-up email, and some information about biocrust. And sounds like really interesting research from your students and your lab that's yet to come. To kind of wrap up, where can folks find you, your research, information from your lab, to stay up to date?
Yeah, you can email me anytime. I'm just brooke.osborne@usu.edu. You can look us right up. I've had it on my to-do list to make a lab website for 6 years, maybe. So I don't know if that's going to happen anytime soon, but USU, I think, does a really good job of sharing and publicizing the findings. And we are fortunate to get a lot of help with that. I think because Moab's pretty, and I think they like to come down here too to make videos and stuff, which I understand. And so, USU, you can watch the newsletters from there, and email me anytime if you're curious, because I do hope in the next few years a lot of these projects will be coming to fruition surrounding this question of dryland soil carbon.
Awesome. Thank you. We'll share if there's any articles that we can pull, or how to sign up for those newsletters. There's really great, nice to read, beautiful newsletters from folks at USU that I get and love looking through. And there's so many interesting things that researchers are doing. But we want to thank everyone so much for attending, your wonderful questions. Thank you so much, Dr. Osborne, for joining us tonight and sharing all of this information. It was super fascinating. Really appreciate your time.
Yeah, my pleasure. Thank you for being here. Thanks for your questions.
Awesome. Thanks, everyone. Well, we will send out the recording. We'll send out contact information, our survey. Again, if you are able to take that, that just really helps us serve our community and what you're looking to see, what you're looking to learn, how we can improve our adult education and walks, talks, and workshops. And you can always reach out to myself or Hunter directly as well if you have an idea or feedback or anything like that. So we'd love to hear from you. But thank you all so much, and have a good rest of your evening. And hopefully we got some winter on the way this weekend.